episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor. Welcome, everybody, again to another episode of our show, bringing you another fascinating guest today, helping to create a better tomorrow on many different fronts for us. Today, we have the honor of being joined by Dr. Marina Ezcurra, who is a lecturer in the biology of aging and neurosciences at the School of Biosciences at the University of Kent in the UK. Dr. Ezcurra received her PhD back in 2011 from the Karolinska Institute, and her PhD research at the time was a collaborative project between Karolinska and the Medical Research Council Laboratory of Molecular Biology, Cambridge, where she was studying neural circuits and behavior in C. elegans in the lab of Dr. Bill Schaefer. During her PhD, she identified various extrasynaptic mechanisms by which nutritional status helped modulate nociception. Went on to do a postdoc working in aging with David Gems at University College in London. During her postdoc, Dr. Ezcurra developed various methods to monitor the development of multiple age-related diseases in vivo in C. elegans, ultimately leading to the discovery of a very interesting and previously unknown process termed intestinal biomass conversion. Really interesting mechanism where basically C. elegans intestines break down over time to produce vast amounts of yolk, resulting in polymorbidities and mortality in aging nematodes. This work further served to illustrate how aging and age-related diseases ultimately can be a result of these, wild-type run-on gene functions rather than just the traditional thought of stochastic molecular damage. Dr. Ezcurra's current research group is focused on how host microbiome interactions affect host aging. She has funding from the Wellcome Trust and Royal Society for this work. Dr. Ezcurra is also a trustee board member of the British Society of Research on Aging. A lot of interesting topics to talk about today. Dr. Marina Ezcurra, thank you so much for taking the time out of your schedule to talk to us.
Thank you. Thank you for having me and inviting me, Ira.
It's a great pleasure having you. I want to start things off like we typically do by basically handing you the floor for a little bit, Marina, just to talk a little bit more about yourself. If you could sort of take us back to the early story, where you grew up, how you got interested in science and neuroscience and aging, and a little bit of the early research journey. I think that'd be a great way to start everything off.
Yeah, sure. So I was born in Argentina, in Buenos Aires, in Argentina. My parents are Argentinian, but at a very early age, when I was very young, we went to Sweden and started living there. So I grew up in Sweden and went to university there at the Uppsala University, and was really sort of fascinated by biology and knew very vaguely that I wanted to do a sort of research career in biology. But during my undergrad, I was kind of like struggling to find that thing that was really going to be, my thing. And then at the very end of my undergrad, I took this neuroscience module. So actually I was studying more sort of biotechnology, so I hadn't done a lot of biology. But I took this extra neuroscience module and was absolutely fascinated. Was a fantastic module. I felt so pulled in and excited. And in one of the lectures, the lecturer, Professor Don Larhammer, spoke about this animal, C. elegans, and showed us, the kind of sort of science that you can do to understand behavior and the nervous system in C. elegans. And it was there and then. I was like, I want to work. This is so amazing. I want to work with this organism. At the time, there was nobody in Sweden who was really doing neuroscience in C. elegans. So I started looking around, and because sort of at the end of my undergrad, I had this opportunity to do a 6-month research project, which I could do anywhere in the world, not necessarily at my home university. So I started looking around different labs, and came across the lab of Bill Schaefer, who was then in San Diego in California. And thought that the stuff that he was doing was really interested. So I emailed him and asked, can I come to your lab for 6 months and do some research? So yeah, there I went. I had to take quite big student loans to make this, possible to happen. But that was really my introduction to C. elegans. I just sort of, it was so early in my research career. I don't think I could actually quite realize the potential of working with these kind of organisms fully, but there was something about it that I kind of understood. This is good. This is going to be really cool. And I started doing a PhD with Bill Schaefer in that lab, which was, like you said in the introduction, joint with the Karolinska, but I was physically in Bill's lab. And as I was starting, he got a job offer to go to the LMB in Cambridge. So the whole lab packed up, and we went across the world and set up the shop in Cambridge. So that was quite a sort of shifting in sort of environment, going from Southern California to a place like Cambridge, but absolutely mind-blowing in terms of, like, the scientific experience, the research experience that I had there. As I was, doing my PhD and going to conferences and learning, more about what people use C. elegans for, I got really interested in the biology of aging. As you're well aware, C. elegans has been, a fantastic tool to understand sort of the fundamental drivers of aging. Yeah, I contacted David Gems for a postdoc. I went to see him, and we had this fantastic conversation about, the aging field, its potential moving forward. And it turned out that he had just received a big grant with Linda Partridge, Dominic Withers, and Janet Thornton to do work on a big program funded by the Wellcome Trust to work on aging. So four labs working together from different angles. So yeah, he had a funding for postdocs. So I joined as a postdoc. And again, was in a fantastic environment, because now, so I went to University College London, and we were at the Institute of Healthy Aging, where a bunch of different research groups using different models work on the biology of aging. So I learned lots about, what people do in Drosophila, in mice, yeah, it was a really good experience. So that's what sort of pulled me into the biology of aging, really.
Outstanding. And as I'm reading through sort of the different groups of work that you've done, and we'll get sort of to the microbiome in a little bit as we focus on your current lab. But the first place I really wanted to start, because I remember when this paper came out, it was extremely fascinating. It was entitled C. elegans Eats Its Own Intestine to Make Yolk, Leading to Multiple Senescent Pathologies, 2018. And here, you go into this really fascinating dynamic, once again, in C. elegans, where there is this sort of this wild type program known as vitellogenesis, or the creation of yolk, that, you think about these mechanisms, many we don't know about. You're discovering new ones all the time, that should stop, but it doesn't. And in this particular case, not only is very important at the beginning of life, but leads to this degeneration and death at the end of the organism's life by this horrible autophagy of the intestine. Talk just a little bit, if you would, about this particular work and sort of where we are in sort of the discovery of some of these mechanisms that sort of sit behind the scenes from a lot of the more stochastic molecular damage that we typically see in the news. But I think we really need to focus a lot more on a lot of what's happening behind the scenes and some of the things that you're discovering along this path.
Yeah, so when you talk about, C. elegans and aging in C. elegans, one thing that people often ask you about is what do worms die of? And we haven't been able to answer that question because we don't know. People have been looking at to a large extent at survival curves, so lifespan of worms, different mutations that will for example increase lifespan. But when I joined David's lab, there was sort of not that much knowledge about what is going on sort of morphologically, what diseases do the worms get, what kills them in the end. So that's really what I started working on, and what we started doing was to develop sort of methods to in alive living nematodes, just follow them throughout life and look at what happens inside in their different tissues. People had done this to some extent in the past, but not in a systematic way. So we started looking at sort of several different tissues in parallel and just observing what happens in the worms. And two of the most striking features is that the intestine of the worm sort of deteriorates horribly. Their intestines just kind of shrink down. They go from being like big healthy intestines and they kind of just shrink down to almost nothing remaining. And the other thing that happens is that they completely fill up with fat. The worm is like a bag full of fat. And this fat is related to the yolk, to production of yolk. So what worms, so just to explain a little bit about the biology of worms, they have short lifespans. And early on in adulthood, they produce eggs like there's no tomorrow. In like 3 days, they lay between 200 and 300 eggs. It's biologically, it's a huge sort of effort that they put into all this sort of reproduction. And what this work sort of eventually led to was an understanding that what the worms do is that they made, in order to produce all these fat that they need for fat and proteins that they need for the egg production, they start breaking down their own intestine to generate this biomass. After 3 days, the worms stop producing eggs. The reproductive period is over, but the production of the fat and the protein just continues. It's like a tap that has been switched on, and there's no evolutionary pressure to switch it off because the worms have stopped reproducing. So what ends up happening is that fat and protein has nowhere to go and just fills up in the body of the animal. So that was, and together with another postdoc in the lab, Alexandra Benedetto, we discovered that this process involves autophagy.
Sure.
And that was quite controversial because we know that autophagy is actually something that's really good. It's a way for the cell to recycle material, in particular, say, for example, during starvation. But in this particular specific situation, the worms are using autophagy to break down their own intestine, so causing actual damage to their own organ. And then that results in all this sort of fat and protein being produced and filling up the body of the animal, making the animal basically obese. So that was what that paper was about, really. So it was quite difficult to publish it. There was sort of, we felt there was a bit of resistance within the field to put this forward, but with a lot of work, we got there in the end. So if you then want to relate this to the biology of other animals, which don't have this specific process of breaking down their intestines to produce yolk for offspring that does no longer exist.
Sure.
What this work demonstrates is more a principle of the kind of drivers that could be driving the aging process, which is that you have sort of biological processes, biological mechanisms that are beneficial earlier in life, for example, to reproduce or to grow, like insulin signaling, that are switched on early in life. And then after, years and years and years of being switched on, they are no longer needed, but it's like a tap that has been switched on and there's no evolutionary process to switch it off. And that links into, bigger theories of aging, antagonistic pleiotropy. For example, the idea that genes can have beneficial effects in some circumstances, but detrimental effects in other circumstances. And the hyperfunction idea that has more recently been put forward by Blagos-Cloney. So that was a study that kind of shows that aging is not necessarily the cause of aging is not necessarily the accumulation of stochastic or this stochastic damage. That might happen as a side effect of other processes that are really have been selected for because they are beneficial early in life.
Sure. I'm glad you pointed that out, because that was one of the things that I wanted to sort of highlight, which is great that you did. The other that for, I guess, listeners that may be less familiar with something like C. elegans and just nematode, whatever. But here, I wanted to also highlight sort of this very unique sort of systems biology that you can actually glean from working with a species like this. So that's why I was equally excited to talk about that. But now, sort of moving from that and bringing C. elegans with us, we move on now into your path into the human microbiome. And now here is a topic we've touched on a little bit on the past. We discussed we have these trillions of organisms living in, on, or in some cases symbiotically part of us. And your work here at the lab really brings together several interesting things from the past. So you're bringing together C. elegans, your work in neurology, because you're now going to be connecting the gut microbiome and the brain axis, neurochemistry, behavior, psychiatric disorders, and aging, of course. Take us a little bit in sort of where you're going with all this, because obviously the microbiome is a very hot topic. You're now bringing in some very exciting components of that, the CNS, your innovative models, and of course, aging. Walk us through a little bit what you're doing here.
Yeah, so over the last, 10, 15 years or so, we've suddenly learned a lot about the microbiome.
Yep.
Because we now have, so earlier on, say when I went to school, I think I was told that the human gut has 200 species of bacteria or something like that. And the reason, and now we know that it's many more.
Right.
And the reason that the sort of the microbiome was sort of underestimated was because what people were doing was they were taking, say, fecal samples from humans or animals and growing any microorganisms that were present in that sample in a lab using standard sort of microbiological techniques.
Mm-hmm.
Now the problem is that many human or many microbes that are present in humans and animals wouldn't grow under such conditions.
Yep.
They can't tolerate high levels of oxygen. They might need very specific, so say media. They might have to feed on mucins from humans.
Mm-hmm.
So a lot of these bacteria didn't really grow. And were therefore not studied.
Mm-hmm.
But then what happened was that as sort of high throughput sequencing techniques, were used more and more widely, became, available and, less expensive, people started actually sequencing what was present in fecal samples. And I mean, it's mind blowing. It's such a diverse set, of organisms that we have present in our gut that now have been are starting to be characterized based on their genome.
Yep.
So that, so suddenly, the scientific community became aware of that we have actually a very rich and very diverse microbiome. A lot of funding and projects was put into looking at it in different aspects, from different aspects. And one thing that people within the scientific community did was that they started, take samples from fecal samples from different patient groups. Mm-hmm. And compare what, if you have a healthy sort of cohort that has Alzheimer's disease or, cardiovascular disease or cancer.
Mm-hmm.
And these studies started showing that actually there can be a lot of differences in the microbiome between these, between healthy and unhealthy individuals. And there are, numerous studies showing this. And that's the kind of studies that over the the last years have gotten a lot of, you see a lot about it in the media.
Sure.
People are starting to become much more aware of the possibility of the microbiome having effects on health. There's a lot of sort of interest, but these kind of studies don't show that if the microbiome actually contributes to disease.
Right.
Or if changes in the microbiome might be caused by disease, or whether there's not really necessarily a relationship. So it's really important to now start to study this in the lab. And and you can do so. So there has been quite a few studies where people have done it in mice. So you take, laboratory mice, you can sterilize them, for example, and start introducing specific bacteria, and study that in a lab environment. But it's quite, it's challenging because you have to have very specific facilities to have like sterile mice. It's expensive, it's slow. So there's really a need to, in addition to say mice, also start working with many other different models that can like just help us understand what are the sort of what is the relationship between the microbiome and health. What kind of interactions do we get between microbes in the gut, and different aspects of host physiology. Myself and other people started thinking, well, C. elegans could be a really good model for this, because it's very easy to work with, inexpensive. You can actually sterilize it very easily just using household bleach. You can then do all kinds of different sort of experimental work that you would want to do in a systematic way. And then people have also started working with flies, looking at using flies to study the microbiome, fish. And that's really where I think what the field needs is a lot of people working using different models, and really, because we need to understand really the basics of sort of how these interactions actually work. So that's what I kind of set out to do, as I was starting up my own lab, establish model systems that are easy and inexpensive to study interactions between the microbiome and the host.
How, obviously, as you're pointing out, the microbiome is a very diverse place, and we have obviously the big one in our gut, but we have all sorts of other ones. There's a urinary tract microbiome. I think there's microbiome in our lungs now. I always see, I see a lot in the press about we're finding new microbiomes everywhere. We're really just a puppet of the microbiome when you think about it. But when coming back to sort of your specialty in connecting microbiome, neurobiology, and aging, is it only the gut that microbiome you're focusing on, or you're focusing on other microbiome? I'm not exactly sure how, where the main, I know the gut brain axis, but are there other microbiome axes in the human body that also are interestingly connected to aging? Right, that's a silly question, just shut me down.
Yeah, so I mean it's a really good question, and that the simple answer is that most of the understanding we have is comes from the gut microbiome, because that has been the most studied. But we also have skin microbiome, vaginal microbiome, and so on. It's just that at the moment, I would say we know much less, and the microbiome is also the sort of the biggest one. So you have to start somewhere. And the other thing to add to that is that when when we talk about the microbiome in general, we talk about bacteria, but actually there are other microbes, there's viruses and so on. But I would say that this is like such a new biological question that we're starting to look at, that you just have to start somewhere, and you start sort of simple. But yeah, I am sure that we, in the future, will learn lots about other microbiomes as well.
Got it.
When it comes to sort of the connection between the gut microbiome and aging and the brain, so there have been some studies, looking at in humans how the microbiome changes with age, and sort of to be very sort of general, what they show is that as we so we're born, we're pretty much sterile. Then as young infants, we start getting exposed to bacteria. They colonize the gut. We start getting more and more microbes and more different species. By adulthood, it has kind of stabilized. And then as we get older, we start getting a less diverse microbiome. So we have fewer bacterial species in total. And the studies also seem to suggest that there is an increase in sort of the more pathogenic species, and a decrease in what's regarded as beneficial species.
Got it.
And sort of related to that, there is also a lot of changes going on in the immune system as we get old. So our immune system becomes less functional. We are sort of less resistant to pathogens. We also, get a more kind of like sort of hyperactive immune system. The immune system is no longer responding as it should.
Got it.
So there's like a really sort of intriguing sort of possible connection there, that there are sort of changes in the immune system, changes in the microbiome, possibly like a sort of negative spiral of things, of that interaction between the immune system and the changing microbiome that might then sort of affect health, other parts of health as we age, including health of the sort of the nervous system. I mean, there are studies in for example in mice showing that changes in the microbiome can affect different aspects of brain health, not only in old people, but also in young people, connections to autism and social behaviors, connections to mood, say depression, and also to neurodegenerative diseases related to aging. And there's lots of super exciting, fascinating research going on where people are starting to figure out the specific mechanisms by which bacteria could be affecting the nervous system through, for example, secretion of specific bacterial molecules, neurotransmitters. Bacteria can actually make neurotransmitters that can then affect the host nervous system, but also through immunity. So bacteria in the gut and our gut immune system are constantly interacting. The job of that immune system is to allow good bacteria to thrive, but to make sure that there's no that keep check of pathogenic bacteria. So there's a constant sort of interaction there, and change. So it's changes in the composition of the gut microbiome can very well be having effects on the immune system, and that can then affect the nervous system through communication between the immune system and the nervous system. So there's lots of super exciting research going on there, C. elegans is a very useful model for that, because because we have a very simple nervous system, super simple, 302 neurons. All the neurons, are mapped. We know exactly where they are. We know to some extent what they do. We can control the bacteria. So we can, put worms on with very specific bacteria, and really get into the sort of nitty-gritty stuff of looking at molecular interactions between the bacteria and the nervous system, and aging, which is sort of what I'm doing.
Yep.
So yeah, that's the beauty of the C. elegans model, the kind of like simple experiments that you can do to look at sort of very mechanistic aspects of these kind of interactions.
And it's clearly a very fertile ground for exploration now, and there's so many directions you can go, which is extremely exciting, as you were saying, between the different microbiomes, the virome, thinking about that, and then obviously the different directions here, the pathogenic, the good, so there's a lot of work ahead of you on this one, so that's exciting. Related to this, and when you were talking about not just the organisms themselves, but the very interesting sort of cocktails of substances that microbes secrete and so forth. I was recently reading about I guess an award you got, or a grant from the National Biofilms Innovation Center, working with a couple other groups. And this is another sort of a couple careers ago, I sort of had an introduction to biofilms, and it's one of those areas that, here we're talking not just about the microbes, but they secrete all sorts of interesting sticky, gluey-like substances in this extracellular matrix, and they mess up a lot, not just the human body, but they mess up a lot of stuff, from ships to bridges. It's trillions of dollars worth of potential if you can deal with biofilms. You're obviously not focusing on all that stuff. You're looking at wound care, Pseudomonas. Talk a little bit about this recent relationship, and then I was just wondering, you're thinking about you're going to be focusing on wound care, but it got me thinking about how these biofilms, once again, do we have biofilms growing inside us with our microbiome that are possibly, as I said, you were saying, messing with some of the systems, and we this is another fertile area of exploration, I guess we should be getting into.
Yeah. So this grant that I got for two industrial partners is a small grant, and the main what we're going to do is actually to develop tools where we make transgenic bacteria that will change color when they produce a biofilm.
Yeah.
And C. elegans is a transparent organism. It's completely transparent.
Yeah.
So the idea is that we'll be able to follow these color changes in living animals as the bacteria in the gut are making biofilms. And in this project is for a very specific sort of context that has to do with, as you say, pathogenic bacteria. But what, potentially in the future, then we can use similar approaches to look at sort of gut bacteria, and be able to really monitor as they form in C. elegans. And why that is important is because we actually know very little about biofilms in humans. So bacteria, as you say, they produce these sticky, what they make is really a film. And they go from being sort of an organism that lives, as a single organism, to becoming like a community. So they can, in these biofilms, the bacteria live as a community. And that kind of changes a lot of the biology and the properties of these bacteria. And there is a lot of interest in understanding to what extent biofilms are formed in our gut, and what kind of effects it has on the biology of the host. But it's so difficult to study that in a human, or even
Sure.
in a mouse, because the films are forming in that gut. It's very difficult to kind of follow that. So there are, approaches using cell culture, but we really don't know how that, translates into a real gut. So it would be really nice to develop these kind of tools that allow us to actually look at how biofilms form. And I'm also very interested in biofilms because actually there has been a couple of very nice papers from, for example, the Noodler lab, showing that in C. elegans, biofilms impact on aging.
Okay.
It would be quite interesting to explore that area a bit more and look, how does this, how do the bacteria form biofilms inside the host gut? Exactly how does that, impact on different aspects of health and of aging? And could this be used to learn more about human aging and health in relation to biofilms? Yeah, that's
another very exciting and really uncharted area. So it's Marian, you really have a lot on your plate, but it's a really fascinating portfolio. Anything? I mean, we touched on the previous work in wild type gene function, microbiome, biofilms. Anything else that is going on in the lab, or any presentations, conferences, anything that you want to mention where you're going to be, that we may watch you in the coming months? Please plug anything else that's that I haven't touched on that you might have wanted to talk about.
Yeah. So the sort of main sort of focus of the lab right now, and something that I recently got funding from one of the main funding bodies here in the UK, the BBSRC, is related to really, well, two kind of strands of work. So one thing that we are doing in our lab is that we've taken bacteria that were isolated from worms in the wild, in the soil, by other labs, by the Schulenburg lab. So they went out and got, because nematodes, worms, C. elegans, they live in the soil. So they went and got some of these, wild worms, and checked what bacteria they have in their guts. And it turns out they have lots of bacteria in their guts. These bacteria, to a large extent, haven't been actually studied at all before. People didn't even know they existed until they were published by this lab. So I got very interested in, like, oh, this is like an opportunity to look at, like, sort of more natural interactions between C. elegans and, and its natural microbiome. So we started looking at these bacteria, and one of the things we found was that a specific species of this sort of natural microbiome improves a model of Alzheimer's disease in the worm. So worms don't normally get Alzheimer's, but you can engineer worms to make to sort of have certain aspects of human of Alzheimer's disease. And I could tell you in more detail, but just to keep it simple. we found that these bacteria actually suppress this disease model. And that was really, quite intriguing. And we continued working on this and realized that this bacteria, even if you take it sort of outside the worm, how should I explain this in a little bit better way? So what we were looking at specifically in the worm is the formation of a certain aspect of Alzheimer's disease, which is these senile plaques formed by amyloid peptides that kind of aggregate and destroy neurons. So this suppressed, the bacteria suppresses this in the worm. Yep. But you can take those aggregates and look at them outside an animal in a tube. And this bacteria also suppresses the formation of these fibrils, amyloid fibrils in this setting as well. And we now know that the bacteria are actually actively secreting a compound that inhibits the formation of these fibrils. So what we want to do now is to figure out a bit more of the detail of exactly how this happens. So that's kind of, potentially it would be absolutely fascinating to then start looking at, if this bacteria can do the same in other animals, in flies, in mice, can we learn, what can we learn about, how bacteria might be affecting Alzheimer's disease in humans? Another sort of aspect of this bacteria that we are working with, that we are really interested in, is that we found that they have a really strong effect on mitochondrial biology. So this is like sort of really, it's really fascinating, because what it tells us is that the bacteria in the gut can actually talk to mitochondria in other tissues. Wow. And affect mitochondrial sort of function. And in the case of our project, that affects function of the muscle. So you could potentially see sort of a scenario where your bacteria, your gut microbiome is affecting mitochondria in different tissues, and that's affecting the tissue, the function of that tissue, like say, muscle function, motility, exercise. So that's that's another sort of aspect of our work that is particularly intriguing at the moment.
Yeah, that's extremely intriguing, and when you talk about it, you think, okay, you have so many interesting paths in terms of can we isolate interesting secondary metabolites from this microbe? Can we increase its natural amount in the body? Can create microbiome cocktails that are beneficial. So it's really neat, especially in this area, the amount of directions that you can go. And once again, I love this type of research, because it shows, as much as we love, being the humans that we are, that in many ways we are still sort of this puppet to this amazing system of microbes and viruses and everything else that we got to respect and keep studying, because it's so important for health and aging and longevity. So really exciting stuff, Marina. I just I've really enjoyed listening to you talk about these programs, and really wishing you the best with all this research, moving it forward in 2022 and beyond. For everybody that's going to be listening to this particular episode on our podcast or watching on the YouTube channel. Once again, you've been listening to Dr. Marina Esguerra, lecturer in both the biology of aging and neurobiology, School of Biosciences at the University of Kent in the UK. Marina, I want to thank you for taking the time out of your busy schedule to come talk to us for a little while. Thank you for everything you're doing there and continuing to do. And as we like to say on our show, thank you for helping to create the better tomorrow through this research. Extremely impressive.
Thank you, Ira. Thank you for having me. And yeah, I love your podcast and the work you do. It's great.
I appreciate that. Thank you.